where phage is absent and the bacteria are happily growing. Okay, so this has led to the idea that phages can therefore be used in order to kill bacteria also in clinical settings, and potentially act as an alternative to more conventional antimicrobials like antibiotics, for example, especially relevant given that many bacteria are now becoming increasingly resistant to many of those antibiotics. And this idea is already applied. It's been popular in Eastern Europe in particular, where phages can be bought over the counter in order to treat infections, and sometimes with very good results. But we're still at the stage of applying phages in clinical context where we have very limited knowledge about when it actually works, and the effects that we may expect, and when treatments will fail, what the optimal treatment conditions are, and so on. So there's lots of questions that need to be answered in order to really implement this strategy. But it's a viable alternative to antibiotics, or can be used together with antibiotics. Now, one potential problem that this application of phage is facing is that bacteria can become resistant against phage. So we see that here. At the top right, we see, again, an infection experiment where the bacteria are fully sensitive to phage infection. But then we also notice that very quickly they can become completely resistant to those phages. And there's two main ways in which they can do that. They can either become surface resistant, or they can become CRISPR immune. And then there are additional defense strategies as well. But if we think about this very rapid evolution of resistance against phages, those are the two main mechanisms. And that's what my lab is studying. When do these mechanisms evolve? How does that depend on the environment? What are the implications? So I will be explaining, first of all, what this means. So what is surface resistance? Well, it's the event where a bacterium is changing its surface, really, the cell wall, in order to avoid infections taking place in the first place. Okay, so this is a very efficient mechanism to deal with phage infections. Avoiding being infected seems a very good strategy. And what it often means is that bacteria have to either lose certain proteins that the phage uses in order to attach to the cell, or that they start to express polysaccharides in order to mask the proteins that would otherwise be recognized by the phage. Okay, so this is one strategy. The other strategy is CRISPR immunity. And I guess everyone has heard of CRISPR-Cas. This year, the Nobel Prize was awarded for the application of CRISPR-Cas for genome editing. But, all that work where CRISPR is used to edit genomes is based on the initial discovery that CRISPR-Cas is an adaptive immune system of bacteria. Okay, so this is a more sort of involved mechanism. This mechanism works post-infection. So first of all, the phage is injecting its genome, but then recognition takes place. And this is really a two-stage process. So first of all, bacteria need to acquire CRISPR immunity. And the way that works is that they capture a small piece from the genome of the phage and integrate it into their own genome, into this genetic locus that is called the CRISPR locus. So this is basically a database that contains lots of small sequences from various phages and other mobile genetic elements. And this sequence information is then used later on when another infection takes place in order to defend the cell. Okay, so this CRISPR array is being transcribed into an RNA molecule that is further processed and loaded onto immune complexes. And these immune complexes then can recognize phage DNA simply by base pairing, and they will then destroy the phage genome. So lots of these systems work on the level of DNA, some of them work on the level of RNA. Okay, so what my lab then tries to understand is when will bacteria evolve CRISPR immunity and when will they evolve surface-based resistance? And it turns out that this really depends on the environment. As a model system to address those questions, we use Pseudomonas aeruginosa and its phage DMS3. Pseudomonas aeruginosa is an important pathogen that causes lots of problems in hospital settings, but also in well known to infect the lungs of cystic fibrosis patients. And this bacterium frequently carries a CRISPR immune system, which is shown here at the bottom of the slide. And we asked the question, when does this bacterium use CRISPR-Cas in order to defend itself against phages? And when we think about what can happen in, during an infection, there's basically three potential outcomes. Either the phage will successfully replicate, or the bacteria can acquire CRISPR immunity, or they can use surface resistance in order to defend themselves. And we then run these simple short-term evolution experiments where we mix bacteria with phage and transfer them for a few days, isolate individual bacteria, and examine if and how they evolve resistance. And then we determine the proportion of the different resistance types that naturally evolve in response to phage. And what we then find is that the type of media that we use to culture the bacteria has a massive impact on the type of resistance that evolves. So in high resource conditions, in LB media, we find that almost all the bacteria are evolving surface-based resistance, and hardly any evolve CRISPR-based immunity. But if we then change the media to M9, which is this nutrient-limited, low-resource media, we see that instead the bacteria evolve almost exclusively CRISPR immunity. And we obviously want to understand why that is, and we worked with theoreticians in order to generate this model, which assumes that surface-based resistance is associated with a constitutive cost, a fitness cost that the bacteria pay in order to be resistant, whereas CRISPR-Cas is associated with an inducible cost. So that means that this cost is only manifesting whenever an infection takes place. Okay, and therefore, this inducible cost will accumulate, will become larger if more infections take place, whereas the constitutive cost is fixed. And therefore, the resources in the environment have an impact on the type of resistance that will evolve. Because if there are very few resources, then the host densities will be low. And therefore, phage has difficulties to amplify, and therefore, overall, the levels of phage in the environment will be quite low. And this means that the frequencies of infection will also be quite low. And therefore, an inducible fitness cost, where you only pay the cost whenever an infection takes place, is favored. Whereas under conditions where host densities are high and the phage amplifies very quickly and infection frequencies are very high, it's better to go for a fixed cost of resistance. So it's really, you can think about it as the sort of season ticket option versus single tickets. It's all about the frequencies at which the events take place that determines which one is favored. And, this means that we can now manipulate the type of resistance that evolves simply by changing the amount of phage that is present in the environment. So if we infect with very low amounts of phage, we see that the bacteria almost exclusively evolve CRISPR immunity. If we add more phage to the environment, then we observe that the evolutionary response is changing. But it's not just about phage densities. We found that also the microbial community context has a very important impact on the type of resistance that bacteria evolve. Okay, so look at the green bars here. That is the amount of CRISPR immunity that evolves. In monoculture, we run infection experiments where we just infect Pseudomonas aeruginosa. But then if we add other important human opportunistic pathogens to these infection experiments, we find that some of them can cause a massive increase in the evolution of CRISPR immunity. And if we run an experiment where we mix all those species together, we find that Pseudomonas aeruginosa predominantly evolves CRISPR immunity under those resource-rich conditions. Okay, so what this means is that we identify two important factors here, so phage density and the microbial community composition, that determine which type of defense bacteria will evolve. So why is this important? Should we care about this, or is it not relevant? And we of course wanted to address this question. What are the implications of bacteria evolving these different defense strategies? And first of all, one thing we noticed is that CRISPR immunity has important implications for the ability of phage to persist. So on the left, we see what happens if bacteria evolve CRISPR immunity. We observe that initially phage titers are relatively low, but then the phage amplifies and increases in density. So you see that here. That is because initially all the bacteria are sensitive, but then very quickly those phages are driven extinct. These are 6 different replicate experiments. We see that it's very reproducible and the phage is wiped out very quickly if the bacteria carry a CRISPR-Cas immune system and when they use it. But if we do the same infection experiment with a mutant that lacks a functional CRISPR system, we now see that the phage can persist for the entire experiment, which lasted 30 days in this case. Okay, so the presence of CRISPR-Cas has important implications for the ability of phages to persist. If we think about potential applications in the clinic, then of course, this is not what we want. We don't want phage to be wiped out very quickly. Instead, we want phages to persist for long periods of time. There's a second reason why the evolutionary response is important, and that has to do with the virulence of the pathogen. So it's well established that the evolution of phage resistance is very often associated with virulence trade-offs. So what that means is that whenever a bacterium evolves phage resistance, it basically becomes less virulent. It's very frequently observed. And this is because changing the cell surface also means that the interaction with the host that the bacterial pathogen tries to colonize is changing, and often not in a positive way. So it becomes more difficult for the bacterial pathogen to colonize its host. So we tested whether that was also the case if bacteria would evolve CRISPR-based immunity. And we see that that is not the case. So if we look at virulence here, measured through infection experiments of these wax moth larvae infection models, and we then measure the time it takes for the Pseudomonas aeruginosa to kill these larvae, we find that the sensitive bacteria, phage-sensitive bacteria, kill the larvae after about 36 hours. Bacteria that have acquired CRISPR immunity kill the larvae after about the same period of time. And this is not statistically significant, this difference. But then if we run the infection experiment with a surface mutant that has changed its cell surface and has become phage resistant through that mechanism, we see that it now takes significantly longer for these mutants to kill these larvae. So there is a clear virulence trade-off, but only if bacteria evolve surface resistance and not when they evolve CRISPR immunity. Okay, so being able to manipulate this evolutionary response could be quite important in clinical settings. Okay, so finally, I want to to say something about why not all bacteria carry CRISPR-Cas immune systems. I've just explained to you how great it is to have a CRISPR immune system, because it enables bacteria to very quickly become phage resistant, wipe out the phage completely, and maintain their virulence. And yet we see that approximately 60% of all bacteria don't carry a CRISPR-Cas immune system. And in the case of Pseudomonas aeruginosa specifically, about half of them don't have a CRISPR-Cas immune system. So why is that, given all these advantages that I just explained to you? In order to address that question, 2 postdocs in my lab, Claire Rowley and Ansha Pellegrot, started to run these competition experiments where they take a wild-type Pseudomonas aeruginosa with a CRISPR-Cas immune system, and they mix it with a mutant that has an inactivated CRISPR-Cas immune system. So the delta Cas7. Cas7 is a key component of the CRISPR-Cas immune system. They generate this 50-50 mix, and then they look how the frequencies of these 2 different genotypes change over time and how that depends on the environment. So first of all, in this experiment, you can see that the relative fitness of the 2 genotypes is exactly 1 when there is no phage. And a relative fitness of 1 means that they are equally fit. Then they ran the experiment where they added this phage, DMS3-Ver, to this competition experiment. And now you see that the relative fitness of the wild type is higher than 1, meaning that the wild type is increasing in frequency. So it's fitter than the Delta Cas7. In other words, CRISPR-Cas provides a clear fitness advantage under those conditions. And then they carried out the experiment where they used a phage that can not just go through this lytic cycle, but it can also cause a latent infection. So what that means is that the phage is infecting, but then it's hiding. It's integrating its own genome into that of the bacterial host. It's not doing anything. It's just replicating along with the genome of the host and transmitting vertically from mother cell to daughter cell. So this is very common for phages to be able to do this. And when they infected the bacteria with a phage that can enter into this lysogenic state, they observed that the wild type suddenly had a much lower fitness than the mutant. So it was decreasing in frequency. And lots of detailed analyses showed that this was because those cells that carry these latent infections, they are sick. You can see that here in this experiment already. The lysogens that are being formed are decreasing in frequency very quickly. So this is an infection experiment where initially sensitive bacteria are being infected, and then lysogens are being formed during this experiment. But these lysogens, they decrease in frequency. Whereas in the case of a Delta Cas7 mutant, we observe that these lysogens go to fixation. And then it turned out that indeed these lysogens that are being formed in wild-type bacteria are very sick, shown here as a relative fitness value that is very close to 0 when they compete with a surface mutant, a bacterium that lost the receptor. However, after a few days, this completely disappears. After 5 days, the wild-type lysogens are fitter than the surface mutants. So somehow the negative effects of CRISPR-Cas have disappeared. And it turns out that these bacteria have simply kicked out their CRISPR immune system. So these are some PCR results that all come out negative after 7 days. And this is some resequencing data, whole genome resequencing. And it shows that the CRISPR-Cas immune system has been deleted from those strains. And some bioinformatics analyses show that the likelihood that these events occur is very high in natural settings. We find that Pseudomonas aeruginosa very frequently interacts with these types of temperate phages, and that this can, in many cases, result in those types of maladaptive effects of CRISPR-Cas, and that CRISPR-Cas can therefore be lost very frequently. So in summary, what I've just shared with you is data that show that various ecological factors, including the density of phages as well as the microbial community composition, can impact whether or not CRISPR immunity evolves or whether bacteria use alternative defense strategies. I've also shown you that this has important implications for the virulence of Pseudomonas aeruginosa, and that CRISPR-Cas can drive phage extinct very quickly. But even though CRISPR-Cas provides all these advantages, there are also negatives associated with CRISPR-Cas, particularly when bacteria are infected with phages that can enter into lysogeny. And that's where CRISPR-Cas can be lost very quickly. So with that, I want to thank the people in my lab, as well as funders. I want to thank a couple of people in particular, Stineke van Houten, Claire Rolly, Ansja Villeroe, and Eleanor Elset, who've done the work that I shared with you today, and the wider network of collaborators. And then finally, no, that's it, finally. Okay, thank you very much for your attention.